Afternoon, everybody. How's everyone doing today? Woo, yes, SLAS 2026. Well, thank you for joining our exhibit or tutorial today. This tutorial is going to be on seamless automation from sample management to screening. We are joined today by three of our partners. And for those of you who don't know who HighRes is, HighRes partners with life science organizations to implement intelligent data and lab automation workflows that empower every team member to plan and execute efficient and reproducible science. And today we're joined by three of our partners from Enveda, Heiser, and Merck. The overview for today, we're going to start off with Enveda. We're going to hear from Katie Heiser, who is going to speak on strategically implementing simple and flexible HighRes platforms to accelerate high-throughput screening and annotate the biological functions of nature's chemistry. Now, a little bit about our presenters. Katie Heiser is the director of high-throughput screening at Enveda. She earned her PhD at CU Boulder, where she studied molecular, cellular, and developmental biology. Before joining Enveda, Katie worked for Recursion as Associate Director of In Vitro Pharmacology. At Enveda, she and her team utilizes automated workflows to enable high-throughput identification and validation of hit compounds from Enveda's world-class natural products library. Her team leverages advanced automation to uncover the biological function of nature's chemistry and accelerate the discovery of new therapeutics. I'm going to turn things over to Katie Heiser.
Thank you. Great. So before we get started, I want to tell you a little bit about Enveda and how we do drug discovery differently. So you probably know that a lot of efficacious modern medicines have come from natural sources. On this slide, you'll see a few that are probably familiar to you. Metformin, aspirin, artemisinin, and CBD. These are all compounds that have come from plants, but you can probably think of a few more famous ones that have come from natural sources as well. What you may not know is that natural products actually have improved rates of translation in the clinic. So in this graph on the left that I'm showing you, you can see natural products and natural product-like molecules in green and synthetics in that orange-red color. And as they progress through phase 1, 2, and 3 clinical trials, natural products have actually a close to 50% improved odds of making it to a phase 3 clinical trial. Despite this, we only know less than 1% of nature's chemistry. So this leaves a huge untapped potential. This is why Enveda was founded. So how are we solving this problem? We start with one of the world's largest libraries of natural biomasses. These samples come from plants, microbes, fungus, and they each have thousands of different molecules in them. Our platform is building the world's first chemical sequencer for life. We annotate the structure and function of these compounds, and then when we find a molecule that has the function we're looking for, we give it to our pipeline team, and they use medicinal chemistry and SAR to turn that compound into a medicine. How are we building the world's first chemical sequencer? So on the left, I'm showing you an example of ChatGPT. ChatGPT is translating a poem, a metabolomics poem from English to Hindi. I'm told it's done this quite well. It's very good at translating languages. Very similarly, Enveda has machine learning models that translate the language of mass spec data into chemical structures. This is what allows us to annotate at scale what the structures are that are in all of these natural biomasses. However, we also need to know what the function is of these molecules. That's where my team, Platform Biology, comes in. We're working to annotate the function of nature's chemistry. We do this with fairly standard drug discovery workflows. So we start with single point screening in a primary assay. We use concentration response validations, and then we move our molecules into orthogonal validations, cytotox counter-screening, and additional counter-screens for the programs. However, we also have one additional step. We also screen fractions that can contain semi-purified natural products. Our chemistry team will then isolate any of the molecules that we find that are active, and then we send those through the rest of our screening funnel, through the same validations that the other molecules have gone through. We do all of this in primarily ultra-high throughput. We run most of our platform in 1536 well. But we do enable our systems to have some 384 and 96 well assays as well. This is our platform biology team. There's 8 scientists and associates on our team. We're all biologists. We run in end-to-end platform. So we do all of the assay onboarding, all of our own cell culture. We do data processing, data analysis. And of course, because we're here today, we also run our automation systems. So I want to talk to you a little bit about how we decide what we automate at Enveda. So here in this chart, you'll see your favorite assay. You can find high content screening or reporter assays, viability, add mix read workflows, biochemical or biophysical interactions. These are all types of assays that we run as part of our platform biology workflows. In the color-coded sections of your favorite workflow, you'll find what we've decided to automate at Enveda. So we have 3 work cells that cover compound printing. So those are the steps that are shown in green colors, high content imaging in purple, and our add mix read workflows that are shown in blue. So how did we arrive at these 3 systems? The steps that we focus on automating are the low complexity, time intensive tasks. And we build our systems to have high capacity and they're fit for purpose. The reason that we do this is we want our systems to have a high robustness and low scheduling complexity. We want them to be able to run autonomously without errors. So these are those 3 systems in a little bit more detail. This is our add mix read system on the left, our compound printing system in the middle, and our high content imaging system on the right. All 3 of these systems are also outfitted with webcams so that we can remotely recover and troubleshoot them if no one is present in the lab. And we also work with HighRes to design these systems to be intentionally upgradable as we build them. So to take it one step deeper, this is our compound printing work cell. This is our actual lab. This is the real system. It has 2 major workflows. So the first workflow is a compound pre-printing workflow, where we have both of our assay plates and our compound plates in the ambient storage system here. The assay plates then move to a lid valet to have the lid removed. The compound plates move to an X peel to have them unsealed, and then both of them travel to the echo to do the compound printing in a randomized pattern. Once the printing is complete, the plates are resealed, relidded, and returned to the ambient storage system. Additionally, we recently upgraded the system to handle live cell dosing as well. So when we originally put this system in, we worked with HighRes to leave open space on the system, both physically in the lab and in the design. We recently decided to fill that space with this SteriStore, so that handles our live cells. And we also integrated, although it's a little hard to see here, there's a multi-drop here that allows us to do dispenses on this system as well. So in the live cell dosing paradigm, the assay plates move from the SteriStore to the echo after dosing, and then they can also have something like a disease stimulus added in a time-controlled manner after that. And again, they can be returned to the SteriStore for the rest of their incubation. The simplest of our systems is probably our high content imaging system. This is a very simple A cell that just moves plates from the AmbiStore into each of these 2 IXM Micro 4s. So this allows us to take images into the weekend, overnight, whenever nobody is present in the lab, so we can acquire a lot of extra data that wouldn't be possible without it. And our final system, which is probably our most complicated, but still a very simple system, is our add mix read system. So the workflows on this system all kind of work the same way. Our assay plates start in either the AmbiStore or the SteriStore, depending on what temperature our assay is happening at. They then move through lid handling with either a lid valet or a lid discard if we're going to throw the lid out. There is a multi-drop here that can add our detection reagent. So you wanted to run cell titer glow, that would dispense the cell titer glow. And then all of the plates are returned to their respective storage system. If the assay is happening at 37 or if it's happening at ambient storage, the plates are returned to that for whatever incubation time we choose for that workflow. That could be 30 minutes or 4 hours, depending on the assay. When the incubation period is done, the A cell will then move those plates into our ferristar, which is our plate reader that reads out the endpoint of that assay. So this enables us to do all of our luminescence, fluorescence, HTRF, and absorbance assays on a single system using these very simple workflows. So what I've told you today is that Enveda is translating nature's chemistry into medicines at scale. On platform biology, we do that in two ways, with both pure compound screening and semi-pure fraction screening. We focus on automating low complexity, high capacity tasks. This enables us to support a very wide variety of assays. And we can also support them in ultra-high throughput and mid-throughput using these systems. This allows us to operate a very small, agile team with limited overhead. And we also make sure we enable our systems to be flexible in design so that we can upgrade them in the future and enable new workflows. So that's it for me. Thank you very much for your attention, and thank you to the organizers, especially Drew, Neela, and Tom for organizing this. And I think if you have any questions, we're going to hold them until the panel at the end, but happy to take any questions that you have. Thank you.